spa because this warm water, we were just thinking if we were these octopus, it would kind of be like, you know, sitting in a hot tub or a spa with this warm water around you.
Jennifer, we have a question here. Do these octopus all die after hatching one clutch of eggs?
I believe that is the case. So, but we don't know a lot about how long it takes for them to brood this clutch, and I don't know after they hatch the timing of senescence, but I have someone sitting next to me who's also thought a lot about these octopus, and maybe Annie can help us with that.
Yeah, for this particular species, we can't say for sure how long they brood for because there's never been direct observations. But another deep sea octopus that lives at a similar depth was known to brood for 54 months, which is approximately 4.5 years from the time the eggs were laid to the time the octopus abandoned her spot, presumably suggesting that the eggs had hatched. So that's quite a long time. We have reason to suspect that it would take at least a year for the octopuses at Davidson to develop, but we don't know until we can do some further investigation on it. So lots of things to learn ahead of us.
Yeah, I think that is one of the most exciting elements of what we're doing here is exploring the unexplored and finding new things. I think a lot of time people feel we have seen it all and know it all, and there's so much we're discovering down here now and so many questions that we don't have answers to yet, and how much more is down there for us to explore is just completely mind-boggling.
Right, we had no idea there was this aggregation of octopus here until we just happened to come across them when we were looking at this small feature adjacent to the large Davidson seamount. So we had no idea that we were protecting something so unusual. You know, we've never seen anything like this before in the world, this many octopus all aggregating and brooding. So until you look around, you'd never know what you're going to find.
What is that
So
purple
cool.
stuff?
In the water?
Yeah, what is that?
It's a jelly,
Did we
most likely, or a ctenophore or something. Sometimes,
Yeah, it's quite
Yeah.
purple.
yeah. We saw these
Beautiful.
really beautiful, like bright purple ctenophores on the last dives in Cordell Bank and Farallon. So I'm wondering if their distribution is kind of like, along this coastline.
Huh.
Like, yeah.
Sorry to have missed that. I watched some of those dives, but I did not see that.
Excellent.
It's actually absolutely fascinating how much marine snow is in the water column itself. I mean, we focus so much of our efforts on understanding the biology in the deep sea, but just the water column alone, I mean, it's quite fascinating.
Can you tell us a little bit more about that marine snow?
Yeah, so marine snow is typically aggregations of organic matter that's either bits of material that if you have like a messy eater, so crumbs, and it's also animal waste, so things that the animal consumes and then they don't use for their body, and so it gets excreted back into the water column. And so marine snow is kind of like free-floating organic matter, is one way to think about it.
And a newly emerging technology, something we're working on at UCSB as well, is eDNA, or environmental DNA. And I believe some of the samples that we're collecting, we're going to be looking at that. Very cool. You can sample water, and whether it's waste from an organism or a scale or whatnot, we can look at that, look at the genetics of that, and determine what's in the surrounding environment.
Yeah, if you think about like for us, every day you have hair that falls out of your head, you have skin cells that slough off of your skin, you leave traces of you and your DNA behind. So do the animals that live in the ocean. And so by collecting water samples and then analyzing it for the DNA, we can have a snapshot of animals that were in the vicinity of that water sample in the recent past.
Jennifer, why is it only the recent past that we can look at with those samples?
Well, DNA doesn't last forever in the seawater, so it will slowly degrade and you won't be able to detect it anymore. So I don't know, though, exactly how long we're talking about for eDNA. As far as I understand, it's still something that's actually being kind of studied, is the time frame over which we're really looking at that water sample. And as you were saying, some of this material is going to have traveled from the surface down. Some of it will be organisms when we're at the seafloor that are living in that that area of the seafloor. And so I've seen results of some eDNA work where you're detecting, you know, viruses, and you're detecting whales, and you're detecting fish, and all in one sample. So it's a very interesting new way to look at what lives in the ocean and to collect little parts of them without taking a whole organism.
Thank you.
All right, a shout out to Jason in Michigan, who was with us last year and caught the live discovery of the octopus garden. Welcome back.
All right, welcome back,
Hi,
Jason. Are you as excited as we are to be back to look around? I hope so.
During the last shift, we talked about the front porch of the ROV. Maybe we could go over some of the equipment that's on there. Somebody writes in, they love the little drum on front.
The little drum.
I'm wondering if it's the hockey pucks.
Is it silver?
Yeah, it's probably
Are
the
there 6 of them?
hexagonal. Yeah, the drum set.
Yes.
drum
So
set.
each one of those drum heads is actually, it's a squeezer sample, and it's really neat. It works a lot like a syringe, and so you're only seeing the top part. So you see the white part, and that's like a handle where the ROV's arm or manipulator is going to grab the top of that white part, and then it squeezes it, and that upon squeezing it, it basically actuates a syringe type device. And so through suction, a fluid sample can be collected within 1 second, and so you're collecting an in situ snap of your fluid, and then you can bring it back up onto the ship, and we'll filter it, and then we'll send it off to various labs to be analyzed for geochemistry and some biology analysis to look at the genetics. And there's 6 of them so that we can test at least 3 different sites, hopefully, because you want to try to take replicate samples at each of your site in case one looks a little funny, and then you can confirm it with a second. And if both samples look unique, maybe it's easier to conclude that it's a real discrepancy in your data that can be analyzed further, or maybe it was just sample contamination from something else.
So if everything goes ideally, we will have 2 from 3 spots, correct?
If everything goes ideally, and one of the ROV pilots helped us very much in getting these prepped and working with confidence, and so I'm very excited to see them triggered and some samples collected with them today.
Are you going to be doing the analysis?
I am not. I'm going to send these to 2 different labs. One of them's at Moss Landings, it's the Wheat Lab, and the other's at Texas A&M with the Sylvan Lab. And they'll both be doing their respective analyses. I will be hopefully getting the data, though.
Some of the other things that we have on the porch, you can see there, it's pretty colorful. We have a big orange milk crate, and inside of that is something called an Osmo sampler. And Annie brought that with her and put that on board. Maybe you can give us a little bit of a description of what's inside that crate?
Yeah, so actually that crate is packed full of 2 Osmo samplers. And each, what an Osmo sampler does is it basically creates a salt gradient that causes water to get extracted into this sampler, and it's sample is collected into a capillary tube, and a capillary tube is just a very thin tube. So imagine a coffee stirrer and then even cut that in half. So that's like the diameter of the tube we're working with. And so over time, you end up with basically a record of your seawater. And so in this case, these Osmo samplers are set up. We're going to put the sampler at the bottom. It's not going to come up to the surface with us, and it's going to stay down and collect samples for 13 months. And when you get back up to the when we collect it in a year and we bring it back up to the ship, what we'll have is a record of the water from 2 particular spots, because there's 2 Osmo samplers, so we can stick the samplers in 2 different places. And then we can analyze those basically in a timeline of how the water has evolved over time. And based on other uses of these devices, we will see changes over time related to tides or currents, or not so much currents, mostly tides.
That's interesting, and that's one of the things, you know, that we're really interested to know about this location is that the octopus have to choose a spot to cement their eggs to the rock. And once they choose that spot, they can't move their eggs, but the water that's actively seeping out of these areas, it could be flowing strongly when they choose the site, but then not. We don't know how long that persists. And so trying to get a sense of the variability in the water conditions around these octopus, we'll be able to do that by having these different pieces of equipment that we can leave in place for many months.
You know, that brings up a really good point. Like, we talk a lot about the biology, and definitely I think the majority of the interest on this cruise is about the biology and trying to understand the environmental variables to it. But the site that this is with this low temperature fluid venting in itself is very unique, and there's only been about 3 that have been kind of explored to the point where we can start to really understand what's happening there. And Davidson's the 3rd, so the geological and hydrological science at this area is also equally as fascinating as the organisms that live on it. So there's so much to learn about Davidson still, and hopefully there'll be a lot of dives back here.
Yeah. So this will be our kind of first stab at that longer term view on what's happening with the waters that are moving through these rocks and coming out near these octopus. So in addition to that, those Osmo samplers, there's a number of T-bars that are wrapped in different colored tape, and many of them are temperature loggers, and we have one that's wrapped in multicolored tape, red, green, blue, and that one can also, in addition to temperature, it can measure dissolved oxygen. And if you think about dissolved oxygen, that's telling you the availability of oxygen in the water for the organisms to breathe. And so we also want to know about that, because that can affect the health of the eggs and their development.
Actually, we have 2 of those on here.
Do we?
We have one, yeah, the one in the upper left corner with the black and green tape. That is like a wand. So that's we get to take the ROV's manipulator, will lift it up, and it can stick it in various orifices and get real-time data on the temperature and oxygen that we in the van can see on the screen. And then we have the identical instrument with the colorful handle that Jennifer was describing with the blue and green and red tape. And that's a logger that we will also leave on the bottom of the seafloor to be collected next year with the
Osmo sampler.
And the temperature loggers, the other T handles that Jennifer described.
And how often does it log the temperature or oxygen? The ones that we're leaving on the ocean floor?
I have to double check my notes, but I think we set it for every hour.
Oh, okay.
Because the trick is so the instruments only have a certain amount of battery life, and they also only have a certain amount of data space. And so you have to be able to make sure that for the time that you deploy, that you're going to be collecting data the whole time. And so you're limited to the amount of data that you can collect.
Right.
And one of the tricks for us is to put them in places where we can find them again. Because you can collect all this data, but if you can't get the instrument back, you don't get the data back.
Can you talk a little bit more? One of our explorers out there wants to know how we locate the Osmo samplers after 13 months on the seafloor.
Well, that is a lot of where our pilots and navigators come into play in helping us return to specific locations that we've been to before. Maybe they can help to explain how they record that information or use it to navigate back to the same place later.
Well, when we, if we put something on the seafloor, we always drop a target. So that gets the position of the vehicle as best as we have it from either USBL, which is the acoustic return, or from dead reckoning, which comes off of the Doppler. So that gives us a decent idea. There is some error in that though, so it's not always easy to find things. That would be my response. Do you guys have anything to add?
Yeah, I would just say usually combine that, those observations would work better if that's combined with like a vehicle heading and depth at the same time. This is a bit different because we're looking on a cliff, but if you were out on a flat, you could be on the point where it is, but if you're not looking at it from the right angle, you won't see what you're looking for a lot of the time.
And all those data streams are recorded, so we know what the heading of Herc is all the time, so we can get that information later to help us find stuff.
From a less quantitative perspective, one of the reasons that we use such colorful materials to dress and adorn our instruments is so that when you do return in a year, these things stick out, and especially with the bright lights that the ROV has, it kind of illuminates some of these colors that they do exist in the deep sea. There's a lot of excellent biology with great coloring, but these instruments do tend to stick out, and the sedimentation is so low in the deep sea that you really don't run any risk of these becoming buried in sediment.
We are curious whether or not any octopus will end up nudging them at some point. But
I've seen it happen. If they want to be somewhere, they will get there.
I remember on one of our earlier dives this season, we came across a pink flamingo that looked like it had been there for a few years, and there was someone on the website, and they said, "You found our flamingo." It was from a previous research of a different vessel a few years prior.
That's kind of funny.
It wasn't what we were expecting to find. So I now have a great answer when someone asks me, "What's the strangest thing you've ever found at the bottom of the ocean?" And it would be a pink flamingo.
In an octopus's garden. Mike, we've got a viewer that wants to know what our ETA is at this point. I've lost a little track of time here.
Well, we're almost there. We're at 25, almost 2,600 meters now. We're going to 33. So, what's that?
Half an hour.
Half an hour is right.
About
That's easy.
half an hour till we get to the bottom. Oh, this is so exciting. Another explorer wants to know why we are not using the Niskins this time. I believe we do have them on board, yes?
yes, we do have the Niskins on board, and we are planning to collect water samples with the Niskins specifically for eDNA, as we were talking about just a few minutes ago. So we're going to try and collect those water samples with the Niskins. Over kind of close to some of these locations we've mentioned, Octopus Spa and the Vertical Vent, the Octopus Garden, and then we're of course going to keep one for something else amazing that we're going to find, that we're expecting to find something new that we don't know. So yeah, so we are definitely planning to use the Niskins over the course of this long dive.
On this ROV is neat because the Niskins it's almost like a holster. It's just built into the ROV design. At least when we got on board, it was all set up to go, and so using them it's just so convenient because they're already there.
For those of you at home viewing on the internet, if you go to the quad view, our Niskin bottles are mounted on the port side of Hercules, and they're now in frame, so you can kind of see how they work. They have rubber stoppers on the top and bottom, and when the robotic arm pulls a cable, it releases the stoppers so that they will both close on the top and the bottom. And there's like a bungee cord that pulls the stoppers in to close it off. And then, of course, the water pressure pushes them or keeps them from coming out.
Wayne, I'm sorry, Scott, can they also see the push cores in their quad view right now?
They're on starboard. Stand by.
Push cores are a way that we can collect sediment samples, and we do have one scientist on board who is going to be using push cores to analyze some, I think, organic
persistent organic pollutants.
Yeah,
Yes, in the
sediments.
seafloor. In the sediments, that's right.
So you can see the push cores in the screen that says 7 up there, yeah.
Can they see them?
Yeah,
Yeah.
I've switched.
Oh, viewers, I don't know. I don't think that's
I think
the
they just switched it.
Oh, they did.
I
Oh.
just switched from the Niskins on the port
Okay, got
side
it.
to the push cores on the starboard side.
And so we're getting a tour of all the
So it
equipment
looks like
that we're
we
going to use
have,
today.
do we have 4 of them?
I
Is
think
that correct?
that looks like there's 4.
Yeah.
It's a little washed out, a little overexposed.
Push cores are neat. You stick them in the sediment, and they basically suction, keeps them in place. And so once you take one, you don't want to wiggle it around too much because you don't want to lose the suction that's holding the your pretty pristine record. Sometimes the top gets a couple top centimeters get a little mixed, but other than that, you have a really detailed record of the sediment and how, if you can imagine things getting deposited or put on the seafloor over time, and then they build up, and so you get like a little timeline similar to the Osmo sampler and how that works. And so you can look at how your environmental conditions, or in this case your particular organic pollutants, are changing over time.
Yeah, so if you've never seen a push core, it's a long cylinder that is see-through, so we can actually, and then it has the knob, what you're seeing up there, like kind of the knob's on top, and that's what the manipulator arm will grab, and we push it down into the sediments, and then when we pull it back out, you have this nice tube of sediment, and as Andy's saying, the sediments at the bottom will be the oldest in your sample, and the sediments at the top will be the newly deposited component of your sample. So it's a record through time, and it's we've been talking about the marine snow earlier, so that's a major source of what is settling on the seafloor and building up over time. And along with the kind of shed off cells and dying plankton and other things that are slowly raining down on the seafloor, we also in other places are finding microplastics and other human residues like persistent organic pollutants. In this case, 2 things he's looking at are DDT and PCBs, which originate most often from the land and then make their way into the ocean and then slowly move down to the deep sea.
One of our explorers from Germany would like to know if every octopus species are brooding in big colonies. I know we have a couple of species of octopus off the coast of Santa Barbara, out there in the Channel Islands National Marine Sanctuary. They're both in the same genus, the Octopus bimaculoides and Octopus bimaculatus. They both brood, what's the word, solitarily, although they're around each other, but we don't see them in the aggregations like we see these colonies of the moose octopus down at the seamount. Does anybody know of any other octopus that brood en masse like this?
So when we found these large aggregations, we were kind of blown away because you're much more likely to see octopus who are solitary. You know, you see an individual here, an individual there. And this unusual upside down brooding posture took us by surprise when we saw that as well. Now Annie wasn't with us, but she would have known exactly what she was looking at if she'd been with us last year, because she had seen that same behavior and an aggregation of these octopus off of Costa Rica, right?
Yeah, off
Yeah.
to Costa Rica, about 2,500 miles south of Davidson, a little south
Right.
east, really. But
Yeah, and her grouping that you were studying was 100 plus individuals, right? About?
yeah, the brooding aggregation within like a 25 square meter area, approximate, was 101. But on the outcrop itself, which is a kilometer long by a half kilometer wide, similar to the little feature that we're going to visit, there was 606 observations, and so the octopus population was large. The brooding population was about a sixth of it.
Okay.
And it was just so neat because they're so concentrated, and that really was, it makes one think that there's got to be something more to this fluid story than just coincidence.
Yeah. And so the question was, is this common to see in other species? So it was pretty unusual what you found off Costa Rica. And then right after we had seen all of this, you know, with the Nautilus last year at Davidson, we almost immediately had someone send us Annie's paper describing that off of Costa Rica, which was, I mean, I was like, as soon as I could, I was reading through the whole thing, trying to get a better sense of, okay, this has been seen before, and okay, and how does that compare to what we just saw? But it's pretty unusual to see these large aggregations of octopus.
Yeah, I mean, from a science point of view, it's really neat that they've happened to be in the sanctuary, which is an area that we already protect and we can already go and study. And I mean, just in the past year, there's been 4 visits to this site alone to learn about these octopuses. Whereas at the other site, we were there to understand how the geology and how the hydrology works. And so the observations of the octopuses was extra. That was like bonus material that I was able to study with the help of my co-author, particularly Janet Voigt, who's a cephalopod specialist from the Chicago Field Museum. But this site is great because we can go back again and again and really try to understand the environmental parameters that seem to be attracting these octopuses to the site.
Cool. So I know Wayne was just about to break into these are a few of my favorite things when he was talking about the pink flamingo he found on the seafloor. We've got somebody asking, what are some of the favorite things you guys have found on the ocean floor? So, I need 3 things, Jess. Three
Wow,
of
3.
your favorite things that you found on the ocean floor.
Three favorite things on the ocean floor. Oh, that's a difficult question. I think other people should answer this one and come back to me.
Nice. That's all. Sorry, I didn't mean to catch you off guard there.
No worries.
I have a particularly inspiring one. So you know, they tell you when you're doing research that you really have to be passionate about what you want to do, and you got to really love it, and it's got to be something that you're not going to get, it's not going to be a chore to study really in the long term. And I was looking for my passion for a while, and then I got to visit the seafloor, and just seeing the complexity and vibrance of the organisms in the deep sea, someplace so cold and so dark, and so far away from everything I had really understood about life. Really, I mean, that was it. It was finding my research interest for like the long term. So it's kind of
great.
cheesy, but
That's
it
not
was
cheesy.
my favorite thing.
That's great. That's great. Well, I was going to say that I'm fascinated by flatfish. I worked on them for my doctoral dissertation, and I think they're fascinating. If you don't know what a flatfish is, but you've heard of halibut or you've heard of sole or flounder, these are all those fishes that lay flat down on the seafloor, and they actually have one of their eyes when they're a larva has actually moved physically from one side of its head up and over the top of the head to join the other on the other side, and then they take
So
up residence
crazy.
on the seafloor and have 2 eyes on one side. That's called the eyed side, and then the side they're laying on top of that's, you know, touching the seafloor is called the blind side because there's no eye there anymore. And a lot of them are really good at camouflaging themselves against the seafloor, and I know I might not be compelling you to think so, but they are adorable. And there's many different species. And just recently when we were out with the Monterey Bay Aquarium Research Institute, we were seeing a lot of deep sea sole. And they were beautiful, this deep chocolate brown with light blue spots on them. And even most flatfish, the blind side, has no pigment, so it's kind of a white color. But this was chocolate brown on the underside, which I had never seen before, and beautiful animal, really awesome.
So there were specific reasons. You weren't doing it just for the halibut.
Oh, well done.
Thank you. Thank you very much.
So there's one. Or does that count as 3? Because I love many species of flatfish. No, I'm
That'll
kidding.
work.
I have to think of some other good ones. I mean, obviously, we were completely jazzed by what we found out here last year. So that's definitely a highlight. The Dumbo squid that we saw last year on Nautilus was also really beautiful and pretty awesome to watch swimming. So those are 3 that come to mind for me. Unfortunately, at this depth, we are not going to see flatfish, and I'm sorry, because I wish I could get you as excited about them as I am, but we'll have to do that another time.
We saw quite a few on the last cruises. So that was.
Oh,
that was
yeah,
like
I was very
there were a
happy
lot of-
watching
I was, like,
the
learning
cruises
about that.
last week.
Yeah.
Yeah, there were flatfish all over the
We were
place.
tripping over them. They were everywhere. It was
Yeah, it was
Yeah.
great. Yeah,
All
I was resisting writing into you guys all the time. Cute flatfish. Check out the flatfish.
Can you guys just remind us where you were last week? And
Oh.
what depth you were in?
Mostly, I think we only did one deep dive in Bodega Canyon at like 3,300 meters. But for the most part, we were in much shallow, I think like 1,000 or below, like shallower than 1,000.
You guys
meter
got up
depth
to
range.
like 250 meters near the ends of some
We
of those
did,
dives, right? And
yeah.
that's when these schools of krill were right there at the seafloor. And that was
Yes.
really fun to watch. And you even had footage of some of the sea stars catching krill.
Catching the krill,
Oh, that
yeah.
was so neat.
Oh, that was when you were mapping the Pacific seamounts, right? Was
Oh,
that-
no, sorry.
No,
this
this
was
is
in
a-
Greater Farallones National Marine Sanctuary and Cordell Bank National Marine Sanctuaries.
Okay.
So we left was it this morning, yesterday morning?
Yesterday.
I guess it's officially yesterday morning.
Yeah.
San Francisco Bay, and we headed south, but they left San Francisco Bay and headed north.
Okay.
We just
Sure.
blew through 3,000 meters. So
Woo.
Oh, yeah.
Here we come.
We are getting closer.
To the
Yeah, bottom's about 170 meters off the deck.
And we are descending at a rate of 30 meters per minute.
Quick. How many minutes?
Oh, no, don't make me do math. I was just going to mention that
6
we had
minutes.
said we're in Monterey Bay National Marine Sanctuary.
Just
and
under
then
6 minutes.
there's 2 other National Marine Sanctuaries off of San Francisco Bay area and going north. So we have a plethora of sanctuaries in that part of of California.
I think one of the most amazing things about the sanctuaries, and you talked about it a little bit earlier, is this notion that we are protecting areas that we haven't even gone to yet, and sort of the opposite of what we've done in our terrestrial portion of the planet, where we've explored and then colonized, and then we look back and see if we maybe could have done it different. So I think this is really exciting and cutting edge work that we're doing as we continue to expand our knowledge and mapping of the seafloors.
Right, this section of the sanctuary, the Davidson Seamount Management Zone, was added later than the main portion of Monterey Bay National Marine Sanctuary, in part because we had never been out here before. And so it was only in, I hope I want to get this right, I think it was 2002 that first Monterey Bay Sanctuary, with the help of Monterey Bay Aquarium Research Institute, was able to go down and look at Davidson Seamount for the first time and encountered these gigantic corals and sponges. And that got everyone very interested in making sure that those really old, slow-growing creatures on the seafloor were protected from damage. So it hasn't been protected for all that long, and we're only have visited it, you know, a handful of times. And then this portion of it that we're going to now was only mapped, I think, 2 years ago. So we didn't even know this feature existed until, you know, just over the past few years. So there's other sections of this, the area around David's Seamount that haven't been mapped at fine detail yet. And so that is one thing that we may try to accomplish while we're out here, if time allows, to do some additional mapping and potentially find new targets for exploration in the future.
Very cool.
We only have about 10% of the seafloor mapped. We have the entire planet of Mars mapped. So that just kind of speaks to the difficulty that it is to actually get out and work in oceanic conditions. Limitations due to nature, and then also limitations due to personnel, because when you come out on a boat, you have to bring people. And you can only keep people out on a boat for so long, and then also it requires money, and so you need funding. And oftentimes, if there's not a driven reason to find something, if you don't know what's there, why are you going to necessarily go and look, right? So exploring the ocean is now something that I think is becoming much more common. And hopefully by 2030, we'll have, through the JEPCO program, we'll have a full map of the seafloor, at least at a higher resolution.
Yeah, and you know, we're protecting these areas, but we don't know yet all the things that we're protecting. Ooh, there we go, the fish went by.
Wow.
And we'll do a better job protecting the sanctuaries and everything inside when we know what all those things are. So it's really important for us to get out there and map and then take down these cameras and look around.
Where can we see our depth on our screens?
Oh, pull them up if you do the
other thing, the track link bit.
Thanks. We have our data logger back here, Magda. She's keeping me on my toes with what I need to see.
Just let me know, yeah, and I can put
almost off
HiPAC
50 meters.
if you want to see HiPAC, since you have that.
Thank you.
Let me do that. I think
Science is always a team effort. It's always good to be able to ask people what's going on.
All right, coming back. We don't have nav.
I see India has joined us.
Hello, India.
Oh, hey. Might be my family, or maybe not.
If we're close enough to the bottom, this might be a good chance to get the white balance done.
Yeah, we'll do white balance once we're off the deck once we're like 2 meters off.
Okay. Okay.
Right now we're just getting situated with the vehicles.
No problem. Thanks.
Oh, look at that little guy.
I did not see that.
He's kind of swimming around. Gone now.
Someone's asking if we could install a camera to observe the colonies for a longer time.
It's a good idea. It's a commonly suggested idea. Part of the difficulty of doing that is that video data is huge, and so you would need a lot of battery life to run the camera, and you'd need a lot of storage to collect the data. And so like I was talking about with the temperature probe, how you can only collect so many temperature readings because of battery life and storage. A camera would require much more of that storage. Also,
You need a light source.
You need a light source, and you need, just because you have really low sedimentation, you do have biology that can foul your camera, and so your lens could get covered up, you know, within a matter of a couple weeks. And unless you could have like a little windshield wiper.
More battery life.
Yeah, there are some limitations, although I think eventually we'll get there.
Argus. You can like reset track link. I don't know if that'll something to do. Or try and get Rennie up here, of course.
You know, any of these great ideas that you guys come up with and that we can't do yet, it doesn't mean that they're not possible. And there's certainly things that you personally, like if you're passionate about it, go explore it. Go see if you can make it happen, because that's really how science advances. It's just curiosities and doing what isn't done yet.
With some of these limitations.
Yeah, that's one of the
are
things that I
oh.
share with a lot of the K-12 kids that we work with at the Reef, and that is that so much of this is still brand new, and these ideas and how we can do these things no one has thought of yet. And you know, what did they tell the Wright brothers the day before they flew? They couldn't do it, right? And then they did it. So yeah, I just am so excited about this stuff, because the emerging opportunities for technology and people interested in engineering, and again the biology associated with it, there's just so much going on down here. And you mentioned
Right, so the bearing of that
Mars
wall is
before.
about 300 or something like that?
Oh, I love that view from Argus.
So it should be around 300-ish.
I was going to mention, though, that
Well,
some of
I'm
these
going to have to be here somewhere.
limitations on needing the power to do these
300-ish.
observations
Jess.
has been solved. MBARI, Monterey Bay Aquarium Research
I got it
Institute,
in
now
front
has
of
a
me now on the
cabled
sonar.
observatory so that they can do some of these measurements in the deep sea
We'll come
without
down
the
till we get a
limitations.
visual on that,
Yeah.
and we can do the white balance, and then they can have some time to play with the USBL.
Sure. Coming down.
Yeah.
What do you guys mean when you say the white balance?
So the white balance is there's a white area. It's actually a sticker that is on the manipulator arm, and we put that in front of the camera, and then we program the camera to say, in this environment,
Wow, there
at
they
this
are.
depth,
There's something.
Yeah.
Oh.
this is what true white is. The camera has to learn what white is supposed to look like, so that way it can tell if it's supposed to be green or yellow or red. But it first needs to know what white is supposed to be
Hold that for a
calibrated
second,
to.
Jess.
there.
So we're
Or you can come
calibrating
down
our
from the
camera
meter
to
side.
display
Oh my
colors.
God.
I'm just not going to come down anymore.
Oh my goodness.
Let's find some
minute.
fluids.
So we're
Right.
seeing octopus, all these little balls of purplish white, but then you also see to the left there a cuskeel.
Okay,
Yeah.
yeah, that's what I was wondering.
If you're wondering what the fish is.
So folks, we're going to do the white balance we've been speaking about. We're having a USBL issue as well, so we're probably going to try and get that going before we really get started here.
Okay.
Bear with us for a few minutes.
And USBL is how we position ourselves, so it's important to be able to know. Someone asked how we pick up those temperature loggers after how we find them again.
Exactly.
It's based on that data, so getting that in shape.
Yeah, it's our underwater
Great. Critical.
GPS.
But so let's talk about those octopuses for a second. So they look like little white
All right.
dots.
Oh, and that's okay. That's a grenadier.
And
coming
so
to say
what
hi.
you're seeing is the octopus's beak, which is essentially their mouth, and then their arms are wrapped around their mantle, and so their suckers are
I'm gonna come up.
exposed. And so what you're seeing is basically octopus suckers, a little bit of mantle, and a
Yeah,
beak.
just watching the bubble cam to clear all that.
And it's about the
stuff.
size of a bocce ball or a softball.
Can't really see because of that bar.
Yeah, yeah. Close your jaws before you move around over there.
There's a little polychaete in the Argus view. Oh, no, it's out of view now.
Okay, somebody online is saying that they aren't seeing anything. That was 41 seconds ago. I don't know if are we still on Argus cam or
Those.
Yeah,
Pretty
this
clear
is
here.
these three views are what they have.
so if you're on the quad cam, you can see in the upper port
got it.
How about this?
camera.
So get rid of
there
that
we're
float
zooming in. You
beneath the
can see
armpit.
some octopus down there. Right now we're doing the
That's perfect.
whiteboard.
Thank you.
Yeah.
All right, so let me focus on the stripe. All right. Okay, we're going to lose video for just a minute while I black balance first.
Roger.
Black balancing now. All right, now proceeding with white balance. Okay, white balance is complete. Let's see what the colors look like. Yep, they're perfect.
Roger.
Thank you.
Give him a stone.
Roger.
All right, let's go see what we got. But yeah, I don't know if you can tell or not, but like the orange color of the basket and the tape and the purple, all those colors are a lot more accurate to what they would be on the surface now that we've done the white balance.
They
Wayne, you want
are
to come wide,
poppin'.
please?
Come wide, full wide.
Thanks. Thank you. Angle this a little bit this way so I hit the boxes. All right.
Okay, I'll just come in and get a little closer look and sniff around a bit, but we're still going to have to work on our USBL issue before we really take off anywhere.
Okay.
Okay.
Yeah, can you
Coming
come
down
down
with
a
you.
little bit, Jess?
Yeah.
Do we have the porch light on
Yes,
right
I think
now?
it is on. Yeah. Particle enhancer. No, it's not on.
Oh, okay. It's just a lot of reflection off of the
Yeah. We got a lot of
Science.
special bits here.
a lot of the science.
Thank you guys for bearing with us. We've been doing a little light balancing to try and get the cameras dialed in. Hopefully you guys are seeing a much better picture now of the seafloor.
Wow.
All right.
And the
pretty cool.
octopus there.
What are these darker things?
Sea cucumbers.
Sea
I think.
cucumbers, yeah.
Yeah, sea
Oh, wow, they're
cucumbers.
really big and dark.
Can make them darker.
If you're just joining us, we've just reached the seafloor at Davidson Seamount.
Where's right? It just changed.
There's
Oh.
one octopus over
You're
to the
not an
right
SPU.
that
There we
appears
go.
to be right side up.
Way to the right, the one attempting
yeah.
to fight.
Oh, yeah.
He's
Let's take a look
different.
at that one.
He's
So the ones
different.
that are in this kind of inverted posture with their kind of mouths up and their arms wrapped around them are likely brooding females. Sometimes these individuals that are upright with the arms extended can be males
across
or
from-
females.
Yeah, it's down the stairs. I think it's right.
Across from 80. Zoom in there, video.
Yeah, so if we can get
Okay,
a close-up on
zooming in.
this one, we might be able to determine if it's a male or a female. And to do that, we're going to look at
a little bit less.
the octopus and locate the eyes.
Okay.
And then the arms. If you imagine, if you were the octopus facing forward, the arms that were right off the front, the first one to the right, we count as number 1. And then as we rotate around the octopus like it's a clock, we start counting. And in a male octopus, the third arm, as we count around the clock, looks different than the rest. And so right now I can't quite see that arm.
Looks like a normal arm.
Well, I think that's arm number 2. I see 1, 2. I don't see 3. It might
looks like
be
that
tucked
third's
in
tucked under.
behind. The one that, and it looks like it might be modified, but I didn't get a close enough view. If you're noticing 2 green points or lasers there, those help us to measure. They're 10 centimeters apart, correct?
Yeah, that's
Yes.
right. Yeah.
10 centimeters.
Do you want those on for all these shots, or do you want me to? You want to secure them for a couple shots?
If we could put the lasers on the octopus's mantle for a second, and then we could turn them off, I think.
Okay.
Okay, coming wide.
Let's see. There we go.
There we go.
Hold on.
I got it. There we
you
go.
go.
Yeah.
Okay.
I got a couple pictures.
So I think now we can
Yeah.
turn them off.
Yeah.
Thank you.
Securing them.
Yeah, so okay, the arm that's wrapped up behind,
Mm-hmm.
see how the tip of it is
Right,
shorter
yeah.
and fatter? So that's the hectocotylis, and so that is a male.
Okay,
Oh.
very nice.
So we've got our first male sighting of the dive.
He was easy to spot. He was right side up.
Hmm?
He wasn't upside down.
Right, but we've seen females outside of the aggregations too that are
Mhm.
moving around. If you think about it, females at some point are entering into this area and choosing a spot to lay their eggs. So there should be females that are coming and going from the brooding locations.
Wow.
These are some incredible photos that I'm able to get.
But that we find the male tends to use that third arm differently. You can see it's not being
Right.
used the same way as the rest. It's more protected. It's a male.
Okay, are we okay on this guy? You want to sniff
Okay.
around a bit more?
Yeah, let's keep looking around.
Okay, coming wide.
Thank you.
Yeah. Nice flying, Mike. Nice steady flying. Thank you.
What? Oh, we'll get some much better video later in this dive and the next one when we get all this stuff off the porch here.
That's a good start, though.
One of our internet explorers is commenting that these sea cucumbers that look black are actually deep, deep red. They will completely absorb the green lasers when they pass over them.
Cool.
Big coral.
Yeah.
Look at that.
Yes, some of the other things we're seeing here are corals. As we just said, the sea cucumbers.
Is that one of those black corals?
Yeah.
It looks
it's a
like
black
one,
coral,
yeah.
yeah.
It's crazy that they're orange, though.
And then you see some sea stars above the coral that have many arms that are long. Those are called brazingid stars. Next to that, one of the brazingid stars kind of in the middle of the top of the screen, it looks like there's a squat lobster.
Yeah.
Do you want any zooms on any of this stuff?
Sure, while we're here, might as
Yeah.
well. Yeah.
All right,
Okay.
Wayne, go ahead in there.
Okay.
'Cause not only are we trying to characterize the number of octopus and how they're located, but also the community of animals that live around them and with them.
I had to step out of the van for a second. Can you just remind me, are we at the vertical vent area or are we in one of the other sites?
We're at the vertical area.
Approximately. That's
Yeah,
where we were
kind
aiming
of.
for. When we have our position again, we'll let you know.
Yeah, okay.
Oh, you can really see that black spine to the coral now, and so below all that orange fringe, you can see small black lines on each of those branches. Could you zoom in on the octopus just to the left of this guy?
The little tiny one. Or the one coming into the screen now.
Oh, I was talking about the one brooding at the bottom of the screen.
Okay.
Okay, go in there.
All right, we'll need a little more light. Okay, coming in. That one's kind of tiny.
Do we have lasers? Yeah, we
Oh
have
yeah,
lasers.
so
Yeah,
you
like-
can see eggs, I think, right
Let
there.
me
You can
come
see
wide
eggs, and you can
so
almost see a
the
little
lasers.
shimmering on the left, I think.
Okay.
Oh,
Oh, you
uh-huh.
do.
Yeah.
Tell. So you want to take out, well, I don't know, are we, can we use the TO sensor right now, the one that's manipulatable, or is that not?
Well, we kind of can, but there's a very wise plan to try and drop the Osmo box off first.
All right.
I'm not going to say we can't. We can, but it's just a lot. It's very busy on the porch right now.
Okay. And so that shimmering fluid, that's we're hoping to see a lot more of that.
Yeah,
around
just
this
to
outcrop.
the left.
Yep.
of-
You have
Yeah,
to
I see.
just kind of squint to convince
sorry,
yourself
Mike.
you see it, but
No,
But
it's-
you're right. We saw a little of that for sure. Yeah, right there you can pick it up a little bit.
and one way to kind of like, the more shimmery the fluid is, the more temperature
Yeah.
differential it'll have with the bottom water. And so this is probably relatively low temperature because it's hard to see the shimmering. But I anticipate, based on what has been described to me by other scientists, that we're going to see a bit more vigorous venting. And actually what's really cool about this shot, so if you see the octopus, you'll see that green, I know it's kind of hard to distinguish from the brown sediment, but there's a few patches of green oviductal cement on the wall above the octopus.
Right, kind of at 12 o'clock to 1 o'clock above
Yeah.
the octopus.
Yeah, and so those are places that a different octopus presumably has attached her eggs at a previous time. And so this spot has been now being reused as another nursery position. And that's something that we saw at Dorado too. The octopuses did tend to occupy the spots over and over again. And you'll notice it's sediment free there, so that it's like if you had a piece of tape, you wouldn't want to stick your tape to a dirty surface, right? You'd clean it off first so that you could be sure the tape was going to stick. So these octopuses aren't going to attach their one egg clutch, presumably their one egg clutch, to a dirty rock if it's not going to stick. So we think, based on enough evidence, that deep sea octopuses and octopuses in general tend to like to attach their clutches to clean surfaces.
And do you think the venting water also helps to keep those surfaces clean? Like they may have been cleaner than the surrounding and made it partially attractive to the females because maybe that venting water helps to blow away some of the accumulating sediment?
Yeah, we have reason to speculate that that is actually the draw of the venting water, at least at the other site where these octopuses were observed at Dorado, which is the site 2,000 meters from here, so we're not visiting there today. The fluid seemed to have a detrimental impact on the embryonic development, and so we concluded that it's possible that the draw of these sites is that they're sediment-free, related to the fluid discharge.
That octopus that was in the center on the screen near the lasers just a moment ago, look at, I think, can we push in on that? It looks like there's a lot of snails or whelks
It's very
surrounding
senescent.
that one.
Go ahead, Wayne.
Okay, pushing in.
Look at her
Yeah,
eggs are
look
all
at that.
hatched.
Oh.
Gee.
And then the whelks are there probably, they might be there cleaning up. All the, look at that.
I know that there are species of whelks that are predatory. The Kellett's whelk feeds on the swell shark eggs off the coast of Santa Barbara. Is it possible that these whelks could also be feeding on the eggs themselves?
Like the leftover egg casing.
I mean, I was assuming they're eating the egg casing.
Yeah.
If eggs were abandoned and not protected by female, I would think that they might also eat them, as would, we haven't seen them yet, but we have seen in the past quite a few shrimp, and maybe these galatheic crabs are also
Look
coming
at that
in.
shimmer.
So would
You
we
really see it
think
above
this is
this
a female?
octopus. Yeah.
But her eggs are all hatched, so likely she is.
There is quite a bit of shimmer on the top.
Yeah.
on top.
yeah.
This is a good hole.
Okay, coming a little bit wide.
Sure.
But she's probably down pretty low
Just
on
so you can
physical
see the shimmer.
resources at this point. If she has a, yeah.
This is great. Wow, she's really in that fluid.
Yeah.
And then you can see a little squat lobster
Yeah.
hiding in the
To
on
the right. Uh-huh. some brittle stars are in amongst the cracks there as well.
You notice we're not seeing any stars, and we're not seeing any of those little polyps that we were observing last time. This is a very... Hi, Miles.
Oh, he has arrived. All right.